Good morning. It's my great pleasure to introduce Björn Ekvall Memorial Awardee 2023. Björn Ekvall was a pioneer in the field of in vitro toxicology. He introduced already 1983 the basic cytotoxicity concept, which means that most chemicals cause toxicity by interfering with basal cell functions. This concept was later explored by the National Research Council in 2007 in the toxicity testing in the 21st century, a vision and a strategy document. Actually, that was done 25 years later. And in fact, this concept forms the basic for adverse outcome pathway-based hazard assessment. Thus, this microphysiological summit is an ideal place for award ceremony. Björn, to promote and to find out whether the cell culture tests and cell culture in general is how much is used and whether these tests are harmonized, the protocols. He organized and proposed the multi-center evaluation of in vitro cytotoxicity, so MAKE project, which included 50 reference chemicals, 60 non-animal assays, and 100 labs worldwide. In addition to that, he promoted in many ways the development of in vitro tests to replace animal experiments. I met personally once him and got the impression of the very human, friendly, knowledgeable person who was always supporting especially young scientists. During that time I was that sort of person. So I can honestly say that Björn Ekvall was ahead of his time. He was a real visionary. The Björn Ekvall Memorial Foundation was founded 2011 under the Scandinavian Society of Cell Toxicology, and it became an independent trust from 2020. The present board consists of the president, Stina Orensson, who is a professor, Erik Wallum, vice president, professor, I'm the secretary, Dr. Cecilia Clemetson is treasurer, and Björn's son, Professor Carl Ekvall, he's the physicist, a board member, as well as Dr. Karian Gabrielsson Morton. The goal of the Björn Ekvall Memorial Award is to honor the scientific work and the memory of Björn Ekvall by rewarding scientists who have substantially contributed to the field of in vitro toxicology, for instance, developing new in vitro tests and approaches, promoting quality aspects, providing education and training, dissemination in vitro toxicology, and is a frontline scientist. As you see, this is the list of the previous awardees. You might find the familiar names there, and all are really frontline scientists and active, quite many are active today as well. I have a great pleasure to announce the awardee of this year, which is Professor Dr. Marcel Leist, Chair of the In Vitro Toxicology and Biomedicine Development integrated by the Dorenkamp Spinden Foundation at the University of Constance in Germany, and he is also the director of the Center for Alternatives to Animal Testing in Europe. He is awarded in recognition, so he's recognized of his novel and pioneering scientific work, dissemination and promotion of non-animal science with determined and targeted goal to replace laboratory animal experiments in research and also in regulatory testing with high quality in vitro test approaches and testing strategies. So this is very much in line what was the goal of Björn Ekvall. Before Professor Leist will give his talk on the novel models and technologies for developmental and adult neurotoxicity prediction, I want to present a diploma to him, the original of which will come by post, and he will also get a nice sum of euros, which we hope that they are personal for you and which we hope you would use those, something which is pleased to you. Congratulations.
So now we are going to have the pleasure to hear the awardee. So we are going to prepare the presentation, and he has already been introduced, but probably you all know Marcel already.
So thank you again, Tula, and thank you to the foundation. This is, of course, the nicest introduction one can imagine. Just trying to take the microphone. The problem if you are too tall. Good, I think it works like this. So, this is the other side of Germany. For those that are not from around here, that's close to the Swiss border. And that's, thank you, David, where I'm coming from. And of course, I included the same picture, and I think we are very well in line with the achievements of Björn Ekvall, and I feel very much in this tradition, and I think it's very appropriate to sometimes think about the history and about people that had ideas that we are following 40 years ago. Of course, they didn't have the technologies we have now, and so we may sometimes think that these approaches were relatively simple, but they were very advanced at this time. And I was partially involved in some of these follow-up studies. So I'm starting here with a very simple approach. It will not be a classical scientific talk. It will go a little bit around. I'm taking this opportunity of this award lecture to show a little bit what is fun to us and the different aspects of what we have done. And our kind of everyday model is immortalized cell line. It's not a monster cell like we heard this morning, so it's genetically kind of like a primary cell. It's also conditionally immortalized, so the immortalizing gene can be switched off. And then these cells differentiate to post-mitotic neurons. So they have all the typical channels and receptors, they have the electrophysiology, they have intact chromosomes and so on. So this is a model we have been using for a long time, and these are some old figures, but I think they make the point. So we got interested in how intact are the neurites. So we measure the neurite integrity and the viability. This is the offset of the red and black line, and it gives us some specific information whether something is neurotoxic. And just to show you the difference, the compound on the lower right is a proteasome inhibitor. There are several of those on the market. They can give peripheral neuropathies, but this compound would not be identified as a specific neurotoxicant in this assay. And this gives the motivation then also to tune these assays to find better ways, because here the compound simply kills the cells without a specific effect on the neurites. So this is giving you an understanding of kind of things we are looking into, and this model has been used a lot in screens of environmental toxicants for different agencies, in the US and also in Europe, and also for some OECD case studies. So it is a highly validated and model with a high readiness level. But during the talk, of course, we will not stick there with a simple cell culture in 2D. So I will introduce you to further developments. That's what we want to do in this kind of look back to history and then go ahead, and also to some developments that are currently ongoing. So this gives just some examples. So this model can be used for morphological readouts, for biochemical readouts, for functional readouts, characterizing actually a lot of pesticides and other environmental compounds on the market. And again, you see in the example of valinomycin, this is a compound that can kill the neurites without affecting the cell viability. So this would be a highly specific neurotoxicant that can be identified and then further followed up. So another example I want to give is a whole set of case studies in the OECD IATA program. For those that are in toxicology, the abbreviations are more clear. For the others, this is a new concept of an integrated approach to testing and assessment, combining a lot of technologies, and that requires worldwide an exemplification by case studies, and we contributed actually quite a number of case studies, many on mitochondrial toxicity and some others, all centering around the neurotoxicity and using the cells and the model I have just described. And this is very recent, so if you look at the date, so this is an OECD guidance document just passed now, or actually 2 days ago. And this is based on an in vitro test battery for developmental neurotoxicity. So this is the typical area where everybody would say, you can never use in vitro because it's too complicated. It's about compounds that affect the development of the brain in the mother's womb, like during the fetal development. So there are many processes involved, but this is at the moment a frontline area where the first guidance is available on how to deal with such in vitro assays. And this is from the publication which came out before the guidance. This is the test battery, and it centers on key neurodevelopmental processes. So obviously there are many, and there need to be many assays to cover all these aspects. And we worked in parallel together with US colleagues. So in blue you see the European in vitro battery, IVB, and in red you see assays that were gaps in the European battery. Those with the asterisks are assays already established, and now we are kind of trying to complete the whole thing from the US and the European side. And the neurite assays I just described, they have an official name, UKN4, and they are included in this battery. So we get an idea what these assays could be used for, and now I use the opportunity for a little bit of a theoretical introduction. I will continue with data. So if we look at models, we would say this is a 2D. It's not transformed cells, they are human cells, so a next level would actually be something that is human cells. Perhaps a more complex aspect could be mixed cultures. The mixed cultures could be in 3D, and perhaps they could be also perfused and built up as a microphysiological system. So we could say there is kind of a graduation according to test system complexity, just very roughly speaking. And then someone might say, well, but there are very different aspects you need to consider. You know, Björn Ekvall looked at viability, but you could go a step further and look in more details into the phenotype. For instance, the loss of the neurites without loss of viability and so on. You could look at different omics endpoints or even do single cell sequencing in complex cultures. And you could look at many physiological functions, for instance, electrical functions, hormone release, neurotransmitter release, and so on. And of course, even more difficult is not to use the cells in the normal state, but to induce a disease model that models somehow human disease. So you could say, this is according to endpoint or model complexity. So if we choose models, perhaps we need to choose somehow in a two-dimensional space where in the model complexity and the endpoint complexity are we situated. Again, this is a simplification. There is not just two dimensions. So one dimension would be we could look for organ interactions and how much they are reflected in such a model. Or we could go according to the throughput the model offers. Or the level of quality controls, validation and readiness level. So this is another dimension where these models might differ. Or we could be interested how well are biological niches represented. Or we could think about longevity-related issues. Can we study regeneration, resilience? So again, another graduation would be there. And of course, costs is not a trivial point. So we have a multi-dimensional space to select the models. And the question is there an optimal solution in this multi-dimensional space? And I think this fits into the topic of this congress, but also in vitro toxicology. And of course you can say yes, if your personal liking is the main criterion. Because you will have your baby you like most, the model you have developed, and of course everybody can weigh these criteria in different ways, and then you're right. But you could also say no, because in toxicology the best model is the one that does the job. And that is often kind of judged better by others. This is what we call validation. So you could say for each question there's another solution optimal, because the requirement of each question are different. So sometimes a simple 2D model might do. Sometimes a 3D organoid is not good enough, because you need one where you need certain endpoints and you can re-challenge the model or other things. So we have a huge spectrum and we need to find our place in this. And of course, if you have a biomedical question, the question determines this. If you have a toxicological question, it depends on the regulation you need to answer, and that determines the model choice. So I have given you a little bit of a theoretical thought on this, so now we continue a little bit with data, and I give you some examples from these areas. So what other endpoints and what other modifications do we have to the models, and how do we move on from there? And probably and I think one of the exciting things is the self-organizing capacity of such structures. We heard in the morning that these structures might start thinking if they are a brain, but they might start, going to the toilet if they are kidney, or dealing with your alcohol intake if they are liver, whatever. So I think we should not stop thinking there. So one step is, of course, to move from 2D to 3D, and these cells can, of course, be used to form organoids, so we can get nice organoids of dopaminergic neurons, so that is the dopamine transporter. And we can also look into the neurites in these organoids, and the trick is to use a small subpopulation, 1 or 2% of the neurons, which are genetically changed to be colored. They express a fluorescent protein. If we would do this with all cells, you wouldn't find anything. But if you do it with a small subpopulation, you can trace the neurites, and you can also look then at neurite growth or neurite degeneration in such a complex structure. So, of course, you don't know whether this is a functional network. Sometimes you don't need to know. If your compound degenerates these neurons, then you have a clear answer. But sometimes there might be a functional disturbance. And we know that these cells can form functional networks. This is triggering of calcium oscillations by ciguatoxin. It's a typical marine biotoxin that stimulates sodium channels, and then these cells, they kind of oscillate and oscillate with a 1 to 2 hertz frequency. So they just go up and down like the entire network, and this is kind of exemplified also here in the organoids. the resolution on this slide is not so nicely, but we see this 1 Hertz, 2 Hertz frequency. This is not a loop. It is really following on, so they know of one another. These organoids are quite big, but they all go at the same time, so they must have some communication, otherwise they would desynchronize. Good, so we can follow this, and we can of course follow modification of such an oscillation, which is a nice readout. We can enhance it, we can dampen it down, we can modulate it. Another different model would be what we call 2.5D cultures. It's a kind of bit colloquial. We also call it neuroid flares. 2.5D is something between 2 and 3. And who is interested in fractal dimensions knows that you can have dimensions between 2 and 3. In this case, of course, it means doing 3D organoids and then plating them. That means they keep some 3D structures, but they are mostly then 2D. And I will give you an impression of this. So we generate the structures and we plate them. And then the interesting feature of this is they get very long neuroids, so they can get 1 millimeter large or even larger, very easily, very reproducible, and all the cell bodies stay in the middle, so there is no migration. And so we can separate, also for biochemical analysis and others, the cell bodies from the neuroids, because the neuroids are around. And an interesting feature is you can study mechanical damage, cut injuries, because this is so big you can just cut apart. Or you can cut out all the cell bodies and just leave the neuroids and see how long the neuroids survive, as in a cut injury. And this is something I can demonstrate, and we established a number of endpoints for this. Did I stop something here? Let me... I don't know whether it's frozen. Good, so one way to look more into detail is to express fluorescent proteins in mitochondria, to use some of these cells, so there's not everything stained, so you can individually follow some neuroids and the mitochondria therein. If we just go back. Just this one, yeah. Can I go back? Okay. So you will not see the... Yeah, let's drop that one. Yeah, that's not the good one, yeah. Good, so you can imagine lots of mitochondria moving around these neuroids. You can follow all the mitochondria, you can set up some software to quantify the speed and the movement, and you can see very nicely like how the mitochondria usually move along the neuroids and how this is affected by neurotoxicants at concentrations that do not kill the neuroids. So you would see all the neuroids are there, but if there's a treatment with certain toxicants, the mitochondria stop moving and they don't go on. Another application is actually these mechanical cut models. So it's very easy to follow because you can take an area where you have all these parallel neuroids, they're all radiating out, we just cut this, and then we kind of follow this by a video, and you see the degeneration in 24 hours. And we study inhibitors of such neuroid degeneration. Here an example is shown, and 5IIQ completely rescues these neuroids, so after 24 hours they don't degenerate, even if they don't have their cell body anymore. And we imagine that in many neurodegenerative disease we have neuroid degeneration, so we are very interested in such inhibitors, and also the mitochondrial function can be restored here. And at least this one is working. So another question is, now we have moved to 3D, 2.5D, can we add other cells? And so we generate human glial cells, in this case astrocytes. We can incorporate them in these organoids. So in green you see the astrocytes, in red the neurons. They go into these spheres. And of course we can also generate microglia, so these are human microglia, and you can also see how they incorporate into these spheres, so we can study inflammation. So this is a nice game and it's really fun looking at that, but the question is, does it solve a question? So can we use it? Does this make anything different? So, and actually glial cells can increase or decrease the toxicity, or they can even trigger the toxicity by themselves in certain states. And I'm not going through all the experiments in interest of time, but we have shown that inflamed astrocytes lead to a neurodegeneration. We also have very good data that astrocytes enhance the toxicity, for instance, of MPTP. This is a typical toxicant causing Parkinsonism in humans, and the enhancement is at least 100-fold if astrocytes are present. But it can also work the other way around, and I showed you the proteasome inhibitor before that can trigger peripheral neurodegeneration, and astrocytes attenuate the toxicity of proteasome inhibitors by 10 to 100-fold. So it makes sense to add some of these cells to get some better toxicological estimates. And to show this, I pick an example from disease biology. So there are many forms of programmed cell death in the meantime. One of the newer ones in the family is ferroptosis, and this is also considered to be important in a number of neurodegenerative diseases. So there are clinical trials running with iron chelators for Parkinson's disease, because ferroptosis is considered to be one form of cell death playing a role. And so we looked into these organoids where the ferroptosis plays a role, and we can trigger this by a compound like erastin, and we see the loss of the viability. And ferroptosis is defined as a cell death that can be prevented by iron chelators. So we see here 3 different iron chelators. One we developed ourselves, which can be used to prevent this form of cell death, and a number of companies are still looking into such agents for Parkinson's disease. And also ferroptosis is a good example to show the importance of the cell composition. So here are the neurons, all neurons with astrocytes, and we can trigger ferroptosis again with erastin very nicely in the pure neuronal system, but this is prevented entirely in the presence of astrocytes. So again, we can define better what the contribution of these different cell types is. Good, this was sticking now with these original neurons. Of course, there are many other ways to generate many other neurons. And one of our interests is sensory neurons or peripheral neurons. They are not so easily available, and we generate them from induced pluripotent stem cells. So they have also been used for neurite degeneration screens because peripheral neuropathies are pretty predominant, and we are moving into the pain and chronic pain area using such models. So here we see a simple readout of the neurite degeneration of such cells. And I'm just giving you examples again on chemotherapy-induced peripheral neuropathy-related models where we looked at pain receptor signaling, and specific dysfunctions triggered by these chemotherapeutics. And with this, I'm getting close to the end. A fun part again, and not completely done, but the question is, if we can make the peripheral neurons, can we perhaps generate whole ganglia from them in a 3D structure? So these peripheral neurons start assembling, and we see these assemblies, and the advantage is we can keep these cultures for a very long time. So again, if there's no biological difference, if there's no toxicological difference, it's still technically a large advantage if one can keep a culture for 4 weeks or 8 weeks, because the incubation conditions can change. And also, if we plate these cultures, again a 2.5D approach, they grow out their neurites especially quickly. So what we saw with the Loomis cells was after 7 days. This is what we see after 48 hours, we get 1 millimeter long neurites, and we can study cut injuries, and we can study chemotherapy-induced neuropathies. And of course, we would like to get an improvement also of the pain signals, and for that we need long differentiation times under reproducible conditions, and probably the 3D structure is the more reproducible signal to get all the relevant receptors expressed. And of course, in the end, and that's what I'm going to end with, is the self-organization. This is something we are interested in several things, but here in this ganglia, it was very interesting to see if we culture them over 6 weeks, 8 weeks, they're not round anymore, but they start taking a shape where all the cell bodies are on one side and where all the neurites that were kind of in the middle in a coil start getting parallelized. And this is the structure we would also observe in a ganglion. Now, the next steps would be to add the glial cells and so on, and to see whether we can generate something like a dorsal root ganglion and then study outgrowth from something like this. I think this is fun part for the next 10 years or so. And of course, I thank you for the attention. I thank again very much the Björn Ekwall Foundation, and I hope you enjoyed the talk.
Hi, so we have 3 minutes for questions, so anyone have any question in the audience? No? If not, I have one question for you. It's related with the DNT battery. I'm pretty sure many people here are developing new systems and they are wondering how they can maybe contribute. And I was wondering if someone has a method, what is the process to incorporate this method maybe into the battery? Is it complicated or?
This is a good I don't know what, yeah, thanks for this. So David was asking a very relevant question, so I showed the slide with this in vitro battery, and in a way it's a community effort. So if more people can plug in, that can get better and better, until we get to the point where it gets very expensive and resource consuming to run the whole battery. Then we need to see which assays are redundant, which additional information is delivered. At the moment, the process is guided by the OECD working group. You're involved, but also PARC in Europe took over on this. So I think it's relatively easy to communicate with people, and then I think planning would be required to do a test run and see whether there's added value of new assays or whether there are false negatives in the previous run that could be detected by the new assays. So this would be the practical approaches. You don't want to have false negatives. If something drops through, you would need another assay that would pick it up to make people feel confident and safe.
Thank you very much. I can keep going. Okay, I'm going here.
Okay, thanks. Your presentation actually nicely showed that this in vitro model suits, in addition to toxicity endpoint testing, also for disease, like this neuropathology and pain model was very interesting.
We have time for one more question.
Thanks, Marcel. Great presentation. I have a technical question. In your Lumos model, when you look into this neurite outgrowth, you said it's only neurites, the cells are not migrating. Whether, maybe I missed the part, but is it changing when you add astrocytes? Do you see then the astrocytes will migrate out, and whether the neurons will also start to migrate out because of the astrocytes presence?
It's a good point, because in the brain, close to the ventricles, there would be specialized astrocytes, they're called radial glia, and they give some guidance for the young neurons to migrate. So we don't know whether this is reproduced. We know when we plate, then the astrocytes migrate out in a kind of a corona around the neuronal cell bodies. But we haven't observed kind of radial glia like feet. So usually the neurons migrate when they have kind of like prolonged structures of astrocytes and they move along there. So it could be a model in itself to study early brain tissue formation if you get this migration. So we believe it's rather at a later stage that the cells are more differentiated, they have intact neurites already.
Thank you very much. Unfortunately, we don't have more time for questions, so thank you very much, Marcel.
Thank you.
Applause.